All right, so this is the first in a series of videos to describe the work of Mendel and how it's led to our understanding of how genetics and inheritance works. So Mendel uh, was kind of the first person to look at inheritance in a scientific way. Uh, Before Mendel, there were people that had looked at inheritance, but they looked at it in kind of a just kind of trying to think of things in their head, and they didn't really do very good experiments, if you will. Some of the ideas before Mendel were the theory of acquired inheritance, and one of the things that they used to support that idea was giraffes. And so they, when they observed giraffes, giraffes feed on the acacia trees, which are those kind of umbrella-shaped trees, and they have lots of thorns except at the very top of the tree. And so they noticed the giraffes were kind of always eating the leaves at the top of the tree. So the thinking kind of went something like this. The reason why giraffes have long necks because they were always stretching to get to the leaves at the top of the tree. Today we would say, well, that's just ridiculous, and it is. But at the time it was one of the possible theories about how inheritance worked. You got traits by acquiring them, not because you had certain genes coding for them. So Mendel, who had gone to this university, had studied science, but then became a monk, began to study inheritance. Now, he lived in an abbey, which is the monastery basically, and there was a garden there. Now, he didn't have a lab, so he couldn't do kind of traditional lab work, but he did have a garden, so he chose to study peas because they were easy to grow. They had easily identifiable traits, and they had easily distinguishable forms of the traits. So he used the experimental method. He used quantitative analysis. He collected and counted lots of offspring and collected the data and analyzed the data and really did a very good job, if you will, of designing and carrying out and collecting data in his experiment. So Mendel bred peas, and we have an example here. First, he created true breeding peas by allowing peas to self-pollinate for several generations until when he would cross-pollinate them, they would produce offspring that all looked identical to the parent. He then took those pure breeding or true breeding parents that he called the peer parental generation and cross-pollinated, took pollen from one and transferred to the other, and he did the reciprocal crosses as well. So he did white to purple and then purple to white and created the F1 generation or first filial generation. And what he found was, in this case with flower color, all of them had purple flowers. And this was true for all the traits. They all looked like one of the two true breeding parents. So he then allowed them to self-pollinate to produce the next generation, the second filial generation. And what he found was different results. So here is the actual data table that shows the traits that he studied and the results that he got and the ratios that he calculated based on those results. So what is all this? How does this all lead to our understanding of inheritance? Well, again, you take your parental, true breeding, purple and white flowers, produce your first filial generation, 100% purple flowers. So where did the white flower go? Did it disappear? Well, no, it didn't disappear because when he allowed them, those purple flowers from the F1 generation, to self-pollinate, the white trait reappears about 25% of the time in the F2 generation, and we get this 3-to-1 ratio. So we know a lot more about inheritance and genetics and DNA and chromosomes than Mendel did. So some of the terminology that we use today is a little different than what Mendel used, but the results are still the same. We know that traits come in alternate forms. In the case of flower color, you got purple and white. We call those alleles. Those are alternate forms of a trait. We know that the alleles are located on chromosomes. The specific place where a gene is located for a particular trait is called its locus. And alleles vary because they have different sequences of nucleotides or bases in DNA. So we have the A, T, C's and G's are the 4 bases, and it's the sequence of those 4 bases that makes up our genetic code. So purple flower allele, the white flower color allele are 2 different variations of DNA at the same spot on the same chromosome. We know that we inherit chromosomes from both our mother and our father. We call those homologous chromosomes. So for each characteristic or organism, we get 2 alleles, one from each parent because we get one chromosome, one copy of each chromosome from mom and one from dad. We know that we are diploid organisms, 2 copies of each chromosome. So it's basically like having 2 copies of an encyclopedia inside of our cells. So what is the advantage of being diploid? It creates variability. It creates things that evolution can work on, allows us to survive changes in the environment by having this variability there. So obviously in the F1 generation, something is covering up the effect of the white flower color. It's not like they blended together, and that was another common characteristic where we thought, or common trait way we thought inheritance worked was what we called blending inheritance, where you had, say, and there seemed to be some evidence for that. With Japanese 5 o'clock flowers, if you took a pure breeding red, a pure breeding white, and crossed them, the offspring were pink. It seemed like they blended together. But with Mendel's results, we didn't see that. We saw purple, we saw white, and all the offspring was purple. So the purple was, something about the purple was covering up or masking the effects of the white flower. So we call the trait when both are present covers up or masks the effect of the other one, we call that the dominant allele. So it creates a functional protein that masks the other allele. Recessive allele is the one that, in essence, makes a malfunctioning protein when they're both present. But another way to look at the recessive protein or recessive allele is that you need 2 copies of the recessive allele to have the recessive trait, whereas you only need 1 copy of the dominant allele to have the dominant trait. So when we talk about, there's a couple terms we use in inheritance and genetics as genotype and phenotype. So this is kind of the difference between how an organism looks and its actual genetics that causes its looks. So the phenotype is the description of the organism's trait. It's what does it look like? How does it function? It's the effect of the genotype. The genotype is the actual genetic makeup of the organism. Does it have 2 copies of the dominant allele, 2 copies of the recessive allele, or 1 of each? So when we go to look at crosses, we represent alleles as letters. So for flower color, we use P because purple is the dominant trait, and we use capital P and lowercase p to represent the dominant and recessive alleles, respectively. So a true breeding purple flower pea's genotype would be big B, big P, big P. A true breeding white flower pea would be little p, little p. And so to illustrate the first cross that Mendel did, it would be big P, crossed with little p, little p. And that led to all the offspring in F1 being heterozygous or having 1 copy of each. So homozygous means 2 of the same. Homozygous dominant would be the purple, true breeding. Homozygous recessive would be the true breeding white. And then 1 copy of each, that's the heterozygous. And in case of Mendel's traits, the dominant allele covers up completely the recessive allele, and you get all purple flowered offspring. And then, again, we get from that initial cross, we allow them to self-pollinate, and you get a 3-to-1 ratio, where obviously with the white flower peas, we know that those are homozygous recessive, 2 copies of the recessive allele. But just looking at the purple flower, you can't tell if it's homozygous dominant, 2 copies of the dominant allele, or if it's heterozygous, 1 of each. So the phenotype, again, is the color in this case. The genotype are the actual genes or the letters that we use to represent the genes. Sometimes we call that. So to predict the possible results from any cross, we can use a tool called a Punnett square. You guys have probably used these before. We put the male's genes or the alleles that would be in the sperm cell across one side, the females down the other side, and then we look where the boxes intersect and fill in the letters, the corresponding letters. So bring the big P down and the little p across in the first box there, and you get big P, homozygous dominant purple, and we can fill in the rest of the boxes, and you can get your genotypes, and from your genotypes you can predict the phenotypes as well. And this goes very well with Mendel's results of a 3-to-1 ratio from the F1 cross. So about 25% we would expect to be homozygous dominant, 2 big P's, or 1 out of 4. 2 out of 4, or 1/2, would be heterozygous, and 1 out of 4, or 25%, would be homozygous recessive. Now that doesn't mean if there's 4 offspring you're going to get this exact ratio. This is predicting the possible results. So just because you can't say, well, we've already had these, so the next one has to be this. That's not the way this works. This is possible results. So when we look at large sample sizes, like Mendel did, we should come pretty close to these results. So the 3-to-1 ratio, that was a phenotypic ratio. The genotypic ratio in this case is 1 to 2 to 1. 1 homozygous dominant, 2 heterozygous to 1 homozygous recessive. Or in the case of the phenotypes, you get 3 out of 4, or 3-to-1 ratio. So phenotypic and genotypic ratios are not usually the same. They can vary. So like I said, the term homozygous means 2 of the same allele. It can be homozygous dominant, homozygous recessive. Heterozygous, 1 copy of each, or different alleles. How that kind of goes with that. So you can have 2 organisms that have the same phenotype, but not the same genotype if they have the dominant trait. So how can we figure out if they have the dominant trait, whether they're homozygous dominant or heterozygous? Well, the way we do that is by doing what's called a test cross, where you take the unknown genotype individual and you cross it with a homozygous recessive and look at the results. And based on the results of that cross, we can determine whether they were homozygous dominant or heterozygous. So here's how the test cross would work. So based on our Punnett squares, if the individual in question was homozygous dominant, then all the offspring from the test cross should have the dominant trait. If, on the other hand, it was heterozygous, we should get different results. We should get half with the dominant phenotype and half with the recessive phenotype.